Think about this. What single molecule underpins, well, pretty much every living process?
Mhm.
It's the silent majority in every cell, and its absence spells the end of life as we know it.
You're talking about water, obviously. H2O.
Exactly, water. But beyond just, keeping things alive, this molecule, which seems so simple, has this whole suite of extraordinary characteristics.
Absolutely. Properties that dictate the fundamental rules of life,
Yeah.
right down at the molecular level.
So are you ready to really understand the power of water? That's what we're doing today.
Yes, let's do it. In this deep dive, we're really immersing ourselves in the intricate world of water and, crucially, pH. Drawing directly from core biochemistry principles here.
Yeah.
The goal isn't just to, scratch the surface.
Right, we want deep understanding, mastery even.
Precisely. Extracting a profound understanding of water's unique abilities, its solvency, its interactions with biomolecules.
How it actually participates in reactions.
Exactly. And the critical concepts of pH and buffering that it governs.
Yeah.
We're really aiming to build a rock-solid foundation in these biochemical fundamentals.
Okay, let's dive right in then. Water's incredible knack for dissolving things. The universal solvent, they call it.
And for good reason.
So what's the secret? What's the underlying mechanism?
It really boils down to the unique architecture of the water molecule itself. Picture it like a slightly bent Mickey Mouse head.
Okay.
The oxygen atom is the face, the two hydrogens are the ears, but the angle isn't 180 degrees, it's about 105 degrees.
Right, it's bent.
And oxygen is much more electronegative than hydrogen.
Meaning it pulls electrons harder.
Exactly. It hogs the shared electrons in those covalent bonds. So this uneven sharing creates a partial negative charge on the oxygen.
And partial positive charges on the hydrogens.
You got it. That creates a dipole, a molecule with distinct positive and negative poles.
Ah, so it acts like a tiny magnet, basically, with a positive end and a negative end.
That's a great analogy, yes. And this molecular magnetism is the key to why it dissolves so many things.
How so?
Because water has this strong dipole. It has a really high dielectric constant.
Dielectric constant. Remind me what that measure.
It's basically a measure of a solvent's ability to weaken the electrical force, the sort of stickiness, between charged particles dissolved in it.
Okay.
Water's dielectric constant at room temp, 25 degrees C, is impressive at 78.5.
78.5. How does that compare?
Well, compare it to something nonpolar, like hexane. Its value is only 1.9.
Wow, huge difference.
Or even ethanol, which has some polarity, it's still much lower, around 24.3.
So this high number for water, 78.5, what does it do?
It means that when you put charged things, like salts, sodium chloride, for instance, into water, the strong electrical attractions between the positive sodium ions and negative chloride ions get significantly weakened.
Because the water molecules get in between.
Precisely. The polar water molecules surround the ions, orienting their positive ends towards the negative chloride and their negative ends towards the positive sodium. They effectively shield the ions from each other.
Allowing them to break apart and disperse. Dissolve.
Exactly. That's why salts dissolve so readily in water.
That makes perfect sense. But water dissolves more than just simple salts, right? What about larger organic biomolecules? Stuff like sugars or amino acids.
Good question. That's where another absolutely crucial property comes in. Hydrogen bonding.
Ah, yes. Hydrogen bonds.
Remember those hydrogens with their partial positive charge?
Mm-hmm.
They have an affinity and attraction for electron pairs that aren't involved in bonding lone pairs on highly electronegative atoms like oxygen or nitrogen on another molecule.
Okay, so a hydrogen on one water molecule is attracted to the oxygen on a different water molecule.
Exactly. That interaction is a hydrogen bond. And water molecules are masters at this.
Yeah.
In liquid water, it's not just isolated molecules floating around.
It's a network.
A dynamic network, yes. Each water molecule is, on average, making about 3.5 hydrogen bonds with its neighbors at any given moment. They're constantly forming, breaking, reforming.
3.5? How can you have half a bond?
Yeah, it's an average over time and across all molecules. It reflects the dynamic nature. They're not permanent covalent bonds.
Right, and they're weaker than covalent bond.
Oh, much weaker. A hydrogen bond might be around 4.5 kilocalories per mole, whereas an OH covalent bond is over 100 kilocalories.
But there are lots of them.
Exactly. Lots of them collectively contribute significantly. This hydrogen bonding network is responsible for many of water's unique properties. Its high viscosity, high surface tension, high boiling point compared to other hydrides.
Okay, so water bonds with itself. How does that help dissolve other things, like that sugar molecule?
Because those other molecules often have the right atoms, too. Many organic biomolecules have functional groups with oxygen or nitrogen atoms.
Like the OH groups on sugar.
Precisely. Or think about aldehydes, ketones, and mydrides. They have oxygen atoms that can act as hydrogen bond acceptors. They can accept a hydrogen from water into a bond.
Okay.
And molecules like alcohols, carboxylic acids, amines, they have both oxygen or nitrogen acceptors and hydrogens attached to O or N that can act as hydrogen bond donors.
So they can both give and receive hydrogen bonds with water.
Yes. This ability to participate in hydrogen bonding, either as a donor, an acceptor, or both, allows these organic molecules to integrate into water's hydrogen bond network.
They can effectively swap water hydrogen bonds for water solute hydrogen bonds.
You've got it. That's what makes them soluble. Think about dissolving sugar. It's packed with hydroxyl groups. Water molecules can form hydrogen bonds with nearly all of them. That's the magic.
Okay, so water's polarity and its hydrogen bonding ability are the twin engines of its solvency. Powerful stuff.
Absolutely fundamental.
Now let's shift gears slightly. How does water actively influence the structure of the biomolecules it surrounds? It's not just a passive backdrop, is it?
Not at all. It's an active player. The interaction with water is arguably one of the primary driving forces shaping the complex 3D structures of proteins, DNA, RNA, even cell membranes.
How does that work? What forces are involved?
Well, we need to think beyond just the strong covalent bonds holding atoms together within a molecule.
Right, those are like the primary structure.
Exactly. But the folding, the shape, the interactions between molecules or parts of molecules that relies heavily on weaker, non-covalent forces,
Such as
the hydrogen bonds we just discussed, electrostatic interactions like salt bridges between charged groups, and also very short-range forces called van der Waals forces.
Okay.
And then there's the big one, often misunderstood, the hydrophobic effect.
Ah, the hydrophobic effect. Water-fearing. It sounds like hydrophobic molecules are repelled by water or attracted to each other.
That's the common idea. But the reality is more subtle. It's really driven by the water itself.
Driven by the water? How?
Okay, think about most large biomolecules, proteins, lipids. They're often amphipathic.
Amphipathic. Meaning,
Right. Meaning they have both parts that, like water hydrophilic, usually charged or polar groups, and parts that, quote-unquote, fear water hydrophobic non-polar groups, like hydrocarbon chains.
okay, like soap.
Exactly like soap or phospholipids in membranes. Now, consider a protein folding up in water. What happens to those different parts?
Well, the charged bits, the polar bits, they'd probably want to be on the outside, right? Interacting with the water.
Precisely. Amino acids with charged side chains like arginine or glutamate, or polar ones like serine, they tend to end up on the protein surface.
And the hydrophobic ones, like leucine or valine.
They tend to cluster together in the protein's interior, hidden away from the water.
Shielded.
Shielded. Same idea with a phospholipid bilayer, the membrane. The charged phosphate head groups face the water inside and outside the cell.
While the greasy hydrocarbon tails packed together in the middle.
Exactly. Away from the water. This arrangement minimizes the unfavorable contact between the hydrophobic regions and the water.
Okay, so they self-organize to hide the oily bits. But why? Is water actively pushing them away? What's the driving force?
It's less about active repulsion and more about water maximizing its own favorable interactions. It's about entropy.
Entropy, disorder. How does
Yeah.
that play in?
Right. Think about water molecules. They want to form as many hydrogen bonds as possible with each other. That's a low-energy, stable state for them.
Oh, okay.
Now, what happens when a water molecule bumps up against a nonpolar hydrophobic surface? It can't form a hydrogen bond with that surface.
Right. Nothing there to bond with.
And worse, to maximize bonding with other water molecules nearby, the water molecules at the interface are forced into a more ordered, cage-like arrangement around the hydrophobic molecule.
More ordered, like an ice-like structure.
Sort of, yeah. More ordered, fewer options for movement and bonding. This ordering represents a decrease in entropy for the water, and thermodynamics tells us systems tend towards higher entropy, more disorder.
So having hydrophobic surfaces exposed makes the surrounding water less disordered, which is unfavorable.
Exactly. So what's the solution for the water?
Get rid of the exposed hydrophobic surface.
Precisely. If the hydrophobic molecules cluster together, they minimize the total surface area exposed to water.
Ah, so one big oil droplet has less surface area than lots of tiny ones.
You got it. When the nonpolar molecules aggregate, many of those highly ordered water molecules around them are released back into the bulk water.
Where they can tumble around freely and make more hydrogen bonds.
Yes, they regain their freedom, increasing the overall entropy of the water. It's this increase in the water's entropy that provides the thermodynamic driving force for hydrophobic molecules to associate in water.
Wow. So it's not that oil loves oil, it's that water loves water more and pushes the oil together to maximize its own freedom.
That's a fantastic way to put it. It's driven by water seeking its most stable, highest entropy state.
That's a much clearer picture. Fascinating. Now, what about the other non-covalent forces you mentioned? Salt bridges.
Right, salt bridges. These are straightforward electrostatic attractions between oppositely charged groups.
Like a positive amino group on one side chain attracted to a negative carboxyl group on another?
Exactly. Or maybe within the same molecule, or even between two different interacting molecules. Their strength is roughly comparable to hydrogen bonds, maybe a bit stronger sometimes, and they can act over slightly longer distances.
And what's their role?
They're really important for stabilizing protein structures and also crucial for binding charged molecules or ions to proteins and nucleic acids. Think about DNA binding proteins needing to recognize specific charged patterns.
Makes sense. And the last one, van der Waals forces. They sound weak.
They are, individually. Van der Waals forces arise from transient, temporary fluctuations in electron distribution around an atom.
Even in neutral nonpolar atoms.
Yes. At any given instant, the electrons might be slightly more on one side, creating a fleeting, tiny dipole.
Okay.
This temporary dipole can then induce a complementary dipole in a neighboring atom, leading to a very weak, short-lived attraction.
So like tiny flickering magnets.
Kind of, yeah. They only work over very short distances, typically 2 to 4 angstroms. Any closer, and the electron clouds start to repel each other. Any farther, and the attraction drops off very rapidly.
If they're so weak, how important can they be?
The key is numbers. In a large biomolecule, like a protein or DNA, there are thousands upon thousands of atoms packed closely together.
So lots of tiny van der Waals interactions add up.
Exactly. The cumulative effect can be substantial. It's like tiny bits of Velcro. One isn't strong, but a large sheet holds tight. They contribute significantly to the overall stability and specificity of molecular interactions.
So it's really a team effort then. All these non-covalent forces, hydrogen bonds, salt bridges, van der Waals, plus the hydrophobic effect working together.
Absolutely. It's the sum total of all these interactions within the aqueous environment that determines the final, stable structure and associations of biomolecules.
Can you give an example where we see all of these playing a role?
The DNA double helix is a perfect example.
Okay.
The two strands themselves are held together by strong covalent phosphodiester bonds. But what holds the two strands to each other?
Hydrogen bonds between the bases. A pairs with T, G pairs with C.
Exactly. Specific hydrogen bonds provide the base pairing specificity. But that's not all.
What else?
The flat base pairs stack on top of each other along the helix axis. There are significant van der Waals attractions between these stacked bases. That stacking contributes a lot to the helix's stability.
Ah, okay. Stacking forces.
And then think about the overall structure. The negatively charged phosphate groups on the backbone. They face outwards, interacting favorably with water and positive ions.
While the more hydrophobic bases are tucked inside, away from the water.
Right. That's the hydrophobic effect in action. Plus, the helical structure positions those negative phosphates as far apart from each other as possible, minimizing electrostatic repulsion.
It really is a masterpiece of molecular engineering driven by these interactions with water.
It truly is. These seemingly subtle forces orchestrate the structure of life itself.
Remarkable. Okay, let's move on to another vital role water plays. It can actually participate in chemical reactions. You mentioned it's an excellent nucleophile.
Yes.
What exactly does nucleophile mean in this biochemical context?
Okay, so many metabolic reactions involve one molecule attacking another. The attacker is usually electron-rich, seeking a positive or electron-deficient center. That attacker is called a nucleophile, literally nucleus-loving.
And the molecule being attacked?
That's the electrophile, electron-loving. It has an electron-deficient atom. Importantly, these don't necessarily need to have full formal charges. Partial charges are often enough.
And water fits the bill for a nucleophile.
Perfectly. Think about that oxygen atom. It has two pairs of unshared lone pair electrons, and it carries that partial negative charge. That makes it ready to attack electron-poor centers.
Are there other important biological nucleophiles?
Oh, absolutely. The oxygen atoms and phosphates, alcohols, carboxylic acids, the sulfur and thiols, nitrogen atoms and amines, and notably, the imidazole ring of the amino acid histidine is a very important nucleophile in many enzyme active sites.
And common electrophiles that water might attack?
Key ones include the carbon atoms and carbonyl groups. You find those in aminides, esters, aldehydes, ketones. Also, the phosphorus atom in phosphester bonds is a frequent target.
So when water acts as a nucleophile, what kind of reaction typically happens?
The most common outcome is hydrolysis.
Hydrolysis.
Okay.
Hydrolysis, breaking with water.
Exactly. Water attacks a susceptible bond, like an aminide bond in a protein, a glycosidic bond in a carbohydrate, or an ester bond in a lipid, and the attack leads to the cleavage of that bond. Water gets incorporated into the products.
So this is how we break down large polymers into smaller pieces. Digestion, for example.
Precisely. Hydrolysis is fundamental to breaking down biopolymers, and the reverse reaction, condensation, where monomers are joined together and water is released, is how they're built.
If water is everywhere in the cell, and it's a good nucleophile, and hydrolysis breaks down these crucial bonds, aren't our proteins and DNA constantly under threat of just falling apart?
That's a really important point. Thermodynamically, yes, the hydrolysis of most of these bonds, like peptide bonds or phosphodiester bonds, is actually favorable. It releases energy.
So why don't they just break down spontaneously all the time?
Because of kinetics. There's a significant activation energy barrier that needs to be overcome for the reaction to happen at an appreciable rate.
The reaction is favorable, but slow.
Very slow under normal physiological conditions, neutral pH, body temperature. These bonds are kinetically stable, even though they're thermodynamically unstable with respect to hydrolysis.
So how does the cell break them down when it needs to, like recycling old proteins?
That's where enzymes come in, biological catalysts.
Right, enzymes speed up reactions.
Exactly. Enzymes like proteases for proteins or nucleases for nucleic acids are specifically designed to lower the activation energy barrier for hydrolysis of their target bonds. They provide an alternative reaction pathway.
Making the reaction happen much, much faster.
Millions or even billions of times faster. But crucially, enzyme activity is highly regulated.
So they only work when and where needed.
Precisely. Through mechanisms like activation, inhibition, compartmentalization within the cell, it allows for precise control. Water has the potential, but enzymes wield it selectively. Think of water as the available tool and enzymes as the skilled workers deciding when and where to use it.
Okay, controlled demolition. Makes sense. You also mentioned water in group transfer reactions.
Yes, many metabolic reactions aren't just about breaking things down, but transferring a chemical group from one molecule, a donor, to another, an acceptor.
Like moving a phosphate group around.
That's a classic example. But water can also be an acceptor. Think about breaking down glycogen, the storage form of glucose. In hydrolysis, a glucosyl group, a glucose unit, is transferred from the glycogen chain to water, releasing free glucose.
Water is the acceptor.
Yes. Alternatively, in phosphorylasis, the glucosyl group is transferred to inorganic phosphate instead of water. Different acceptor, different product.
I see. And you mentioned these are often coupled to ATP hydrolysis?
Often, yes, especially in biosynthesis. Building complex molecules, the reverse of hydrolysis, essentially condensation, is usually thermodynamically unfavorable. It requires energy input.
So the cell couples it to a very favorable reaction.
Exactly. Like breaking the high-energy phosphate bonds in ATP. Enzymes facilitate this coupling, using the energy released from ATP hydrolysis to drive the unfavorable synthesis reaction forward.
An elegant energy management strategy.
Very much so. It allows life to build complexity against the thermodynamic gradient.
Okay, fascinating rules for water as both solvent and reactant. Now, let's tackle pH. We all have a general idea, acid, base, neutral, but what's the chemistry underneath?
Right. At its heart, pH stems from a property of water itself. Its ability to slightly ionize or undergo autoentomization.
Water ionizing itself.
Yes, even in absolutely pure water, a tiny fraction of water molecules will react with each other. One water molecule acts as an acid, donating a proton.
And another acts as a base, accepting it.
Precisely. This forms a hydronium ion, H3O+, and a hydroxide ion.
H3O plus and OH, the ions responsible for acidity and basicity.
Correct. Now, we often simplify H3O+ and just write H+ for the proton, but keep in mind it's always hydrated in solution. It might even be part of larger clusters like H5O2+ or H7O3+.
So it's not just a bare proton floating around.
No, never. But the crucial thing is that this ionization is in equilibrium.
Meaning it goes both ways. H plus and OH are constantly forming and recombining back into H2O.
Exactly. It's a dynamic process. At any given instant, a specific hydrogen or oxygen atom might be part of a water molecule or an H3O plus ion or an OH ion. It's constantly shifting.
So even in pure neutral water, there's always some concentration of H plus and OH ions present.
Yes, albeit a very small concentration. Statistically, the chance of any single hydrogen atom in pure water existing as a free hydrated proton at any moment is incredibly small, about 1.8 times 10 to the minus 9.
That's tiny.
It is, but water itself is highly concentrated, about 55.56 moles per liter. So if you multiply that concentration by the probability,
You get the actual concentration of H plus ions.
exactly, it comes out to 1.0 times 10 to the minus 7 moles per liter at 25 degrees C.
10 to 7 m, and since one OH is formed for every H plus I, its concentration must be the same.
Correct. In pure water, H plus OH equals 1.0 by 10 to 7 m.
And this is where the pH scale comes from, that number, 10 to 7.
Precisely. First, we define the ion product of water, Kw. It's simply the product of the hydrogen ion and hydroxide ion concentrations, KWH plus OH. Since both are 10 to 7 m in pure water, KW7, 10 to 7, 10 to 7, 1.0 by 1014, mole whale at 25 degrees C. And this Kw value holds true for all dilute aqueous solutions at that temperature, not just pure water. If H plus goes up, OH must go down, and vice versa to keep the product constant.
Right, they're inversely related.
Now, dealing with numbers like 10.7 or 10.14 is cumbersome. So back in 1909, Sørensen proposed the pH scale.
Which is logarithmic.
Yes, specifically pH is defined as the negative logarithm, base 10 of the hydrogen ion concentration. pH she cows and like 10H plus 7.
So for pure water, where H plus is 10 to 7 m,
pH is nodulog 10 to 777.
so that's why neutral pH is 7. It directly reflects that 10 7 m concentration.
Exactly, and because it's a negative log scale.
Lower pH means higher H plus sick.
Correct. A pH of 6 means H plus indices 10.6m, which is 10 times higher than pH 7. A pH of 5 is 100 times higher, so a lower pH means more acidic.
And higher pH means less H plus m, more basic or alkaline.
Right. A pH of 8 means H plus 10 to 8m, and since K2 is constant, OH must be 10.6m. So higher pH means lower H plus and higher OH.
Okay, and just to clarify terms, acids donate protons, bases accept them.
That's the Brunstad-Lowry definition, yes, which is most useful here. Acids are proton donors, bases are proton acceptors.
Now, there's a difference between strong and weak acids bases, right?
Crucial difference. Strong acids, like hydrochloric acid, HCl, or sulfuric acid, H2SO4, dissociate essentially completely in water. If you put 1 mole of HCl in water, you get 1 mole of H plus and 1 mole of Cl.
Okay. Same for strong bases like sodium hydroxide, NaOH, complete dissociation into Na plus and OH.
Yes. But weak acids and weak bases only partially dissociate in water. They exist in an equilibrium.
An equilibrium between the undissociated form and the dissociated ions.
Exactly. Acetic acid, the stuff in vinegar, is a classic weak acid. In water, it sets up an equilibrium. CH3COOH plus CH3COO. Only a fraction of the molecules donate their proton at any given time.
And this is important because many biomolecules are weak acids or bases.
Hugely important. Think about amino acids with their carboxyl, COOH, and amino NH3 plus groups. Nucleic acids with phosphate groups. Many metabolic intermediates. They all contain groups that can gain or lose protons depending on the surrounding pH.
Except you mentioned phosphorylated intermediates can have a strongly acidic first proton.
Ah, yes. Some phosphate monoesters, for instance, have a first pKa that's quite low, meaning that first proton comes off very easily, almost like a strong acid. But their second proton dissociation often falls in the weak acid range, near physiological pH.
Okay, so understanding weak acid-base behavior is key for biology.
Absolutely essential. Because the protonation state, whether a group has its proton or not, affects its charge, its structure, its ability to hydrogen bond, and ultimately its biological function.
And it's all dependent on the pH of the environment.
Precisely. Intercellular pH changes can dramatically alter the structure and activity of proteins and enzymes. Plus, the charge state is the basis for techniques we use to separate biomolecules like electrophoresis or ion exchange chromatography.
Right, they separate based on charge at a specific pH. Now, you mentioned equilibrium. We often talk about conjugate acids and bases. Can you define those?
Sure. It's simple, really. When a weak acid, let's call it HA, loses its proton, H+, what's left behind A is called its conjugate base.
KJ, H plus A, okay, is the conjugate base of HA.
Correct. And conversely, HA is the conjugate acid of the base A. They're a pair linked by the gain or loss of a single proton.
So for acidic acid, CH3COOH, the conjugate base is the acetate ion, CH3COO.
Exactly. And for a weak base like ammonia, NH3, when it accepts a proton, it forms its conjugate acid, the ammonium ion, NH4 plus Wal.
Okay, clear. And the strength of a weak acid, its tendency to donate that proton, is quantified by
By its acid dissociation constant, Ka.
Ka. How is that defined?
For the generic weak acid HH++ A, the Chi is the equilibrium constant for this reaction. It's Ka H plus A, HA.
So product concentrations over reactant concentration.
Right. A larger Chi value means the equilibrium lies further to the right, more dissociation, more H plus produced. So a larger Ka means a stronger weak acid.
Okay. But Ka values can be tiny, right? Like the H plus concentrations.
Exactly. They often involve negative powers of 10. So, just like with pH, we often use the pK scale.
Which is?
Pico equals decial log tano.
The negative log. So the relationship is inverse.
Yes. A stronger acid has a larger Ka but a smaller pK. Lower pK means stronger acid.
Got it. Can you give some typical pK ranges for common biological groups?
Sure. Carboxylic acid groups, RCOOH, typically have pK values around 4 to 5. Protonated amino groups are NH3+, usually have pK values around 9 to 10.5.
So at neutral pH 7, carboxylic acids would be mostly deprotonated, RCOO, and amino groups would be mostly protonated, RNH3+.
Exactly. Other important ones. The bicarbonate buffer system involves carbonic acid, H2CO3, which has a first pK around 6.4 relevant in blood. The phosphate buffer system involves H2PO4, whose pK for losing the second proton to become HPO42 is around 7.2, very close to intracellular pH.
That pK value of 7.2 seems important physiologically. Is there a special significance to the pH value that equals the pK?
Absolutely. There's a very direct and useful connection. Look back at the Ka expression. Ka H plus A, HA. What happens if the concentration of the conjugate base A is exactly equal to the concentration of the acid HA?
Then AHA equals 1, so Ka just equals H plus A.
Exactly. And if Ka H plus A, then taking the negative log of both sides gives
pKa, bingo.
the pK of a weak acid is numerically equal to the pH at which the acid is exactly 50% dissociated. Half is HA, half is A.
So if you know the pK, you know the pH where it's exactly half and half.
Yes. And you can even find the pK experimentally this way. If you take a solution of the weak acid and add exactly half the amount of strong base needed to completely neutralize it.
You'll reach the point where HAA and the measured pH will be the pK.
Precisely. It's called the midpoint of a titration.
This seems directly related to the Henderson-Hasselbalch equation, which everyone learns in biochem.
It is. The Henderson-Hasselbalch equation is basically just a rearranged version of the Chi expression put into logarithmic form.
Can you walk through the derivation quickly?
Sure. Start with Ka H plus AHA. Rearrange to solve for H plus Ka, JJ.
Okay.
Now take the negative logarithm, base 10 of both sides,
Yeah.
dash log H plus E key log HA. Substitute pH for dash log plus and pK for sat loga. That gives pH pK log HAA.
Okay.
Now remember that task log Hanks is the same as plus log HA, so we flip the ratio inside the log and change the sign outside.
So pH pK plus log AHA, the Henderson-Hasselbalch equation.
That's the one. It's incredibly useful.
What does it tell us? How do we use it?
It directly relates the pH of a solution containing a weak acid conjugate base pair to the pK of the weak acid and the ratio of the concentrations of the conjugate base and the acid HA.
So if you know the pK and the ratio, you can calculate the pH.
Yes. Or if you know the desired pH and the pK, you can calculate the ratio of base to acid you need to make that solution.
Let's test it. What if A equals HA? The ratio is 1.
Log of 1 is 0. So pH equals pKa plus 0. pH equals pKa, just like we saw before.
Okay. What if there's much more base than acid? Say AHA equals 100.
Log of 100 is 2. So pH equals pKa plus 2. The pH is 2 units above the pK.
Makes sense. More base means higher pH. What if it's the other way? AHA equals 110 or 0.1?
Log of 0.1 is nidic 1. So pH equals pKa. The pH is 1 unit below the pK.
This explains titration curves, doesn't it? When you plot pH versus added base for a weak acid.
Exactly. You start at low pH. As you add base, the pH rises slowly at first, then rises most rapidly around the equivalence point where all the acid is neutralized. But in the middle region,
Around the pK.
yes, around the pK, the curve flattens out. The pH changes relatively little even as you add more base. That flat region is the buffering region.
Buffering. Okay, so this equation is key to understanding buffers.
Absolutely. A buffer solution is typically a mixture of a weak acid and its conjugate base. Its function is to resist changes in pH when small amounts of strong acid or strong base are added.
How does the mixture do that?
Think about the Henderson-Hasselbalch equation again. pH, pKa plus log,
Yeah.
AHA. If you add a little strong acid, H+, what happens?
The added H plus will react with the conjugate base A in the buffer, right? H plus plus AHA.
Exactly. So A decreases slightly and HA increases slightly. The ratio AHA changes, but because it's inside a logarithm, the change in pH is relatively small, especially if you have decent amounts of both A and HA to begin with.
Okay, and if you add a little strong base, OH.
The OH will react with the weak acid HA in the buffer, OH plus HHH2O plus A.
So HA decreases, they increases. Again, the ratio changes, but the log term dampens the effect on pH.
Precisely. That's buffering in action. The weak acid conjugate base pair acts like a sponge, soaking up added H plus or OH.
And this is vital in biological systems.
Absolutely critical. Metabolic processes are constantly producing or consuming protons. Think about cellular respiration generating CO2.
CO2 dissolves in water to form carbonic acid, H2CO3.
Right, which is an acid. Without buffering, our blood pH would plummet. But the bicarbonate buffer system, H2CO3, HCO3, is there to maintain blood pH in a very narrow range, around 7.4.
Are there other important biological buffers?
Yes, the phosphate buffer system, H2PO4 to HPO42, is very important inside cells, where its pKa of 7.2 is very close to the typical intracellular pH. Proteins themselves can also act as buffers.
How can proteins buffer?
Because they have many amino acid side chains with ionizable groups, carboxyl groups, amino groups, histidine benzoyl groups, each with its own pK. Collectively, they can soak up or release protons over a range of pH values.
Okay. And in the lab, we use specific buffer chemicals?
Yes, we choose buffers based on the desired pH range for an experiment. Common examples are MES, pK 6.1, HEPs, pKa 6.8, Tris, pKa 8.3, and of course, phosphate itself.
How do you choose the best buffer?
The main rule is to pick a buffer whose pKa is as close as possible to the target pH you want to maintain.
Why close?
Because the buffer works most effectively has its maximum buffering capacity when the pH is equal to its pKa, where AHA.
And how far from the peak area is it still useful?
Generally, a buffer is considered effective within about plus or minus 1 pH unit from its pKa.
So phosphate buffer, pKa 7.2, works well between pH 6.2 and 8.2.
Roughly, yes. Outside that range, the ratio of AHA becomes too skewed, and the buffer loses its ability to effectively resist pH changes in one direction or the other.
So buffering capacity is highest near the pKa? Makes sense.
And the Henderson-Hasselbalch equation lets you calculate exactly how much the pH will shift when you add a certain amount of acid or base to a buffer of known composition.
Right. One last thing related to this. The net charge on a weak acid molecule depends on pH, right?
Absolutely.
Yeah.
Look at that titration curve again. At very low pH, well below the pKa, the weak acid is almost entirely in the protonated HA form. If HA is neutral, the net charge is 0.
Okay.
As the pH rises toward the pKa, more and more HA converts to the deprotonated A form. If A carries a negative charge, the average net charge on the molecule becomes increasingly negative.
Average charge,
Right.
because it's in equilibrium.
Exactly. At pH pKa, half the molecules are HA, say charge 0, and a half are A, say charge -1. The average charge is -0.5.
And well above the pK.
Almost all molecules are in the A form, so the average charge approaches -1. The net charge varies continuously with pH.
And this varying charge is what allows separation by techniques like ion exchange or electrophoresis.
Precisely. By choosing the right pH, you can manipulate the net charge on different proteins or molecules and make them bind differently to charged columns or move differently in an electric field.
Very powerful applications. Okay, final topic. What factors can actually change the pK of a given acidic group? Is it always fixed?
No, it's not absolutely fixed. The pKa is an intrinsic property, but it can be significantly influenced by its local environment.
Like what?
One major factor is the presence of nearby charges within the same molecule.
How does that work?
Imagine a molecule with two carboxyl groups fairly close together. Let's say the first one loses its proton, becoming negative, COO.
Okay.
Now, how easy is it for the second carboxyl group nearby to lose its proton?
It's already near a negative charge. Losing another proton would create another negative charge close by. That sounds electrostatically unfavorable. Repulsion.
Exactly. That repulsion makes it harder for the second proton to leave. So the pico of the second group will be higher, meaning it's a weaker acid, than it would be if the first negative charge wasn't there.
Nearby negative charges raise the pico, make it harder to deprotonate.
Correct. You see this clearly in polyprotic acids like phosphoric acid or citric acid. Each successive pCOV is higher than the last. Conversely, a nearby positive charge would tend to stabilize the negative conjugate base, making the proton easier to lose, thus lowering the pico.
Does distance matter?
Yes, the effect diminishes with distance. Compare the second pico values for succinic acid and glutaric acid. They differ only by one extra CH2 group spacing out the carboxyls. And glutaric acid's second pico is slightly lower, closer to a typical single carboxyl pico, because the negative charges are farther apart.
Okay, so the intramolecular environment matters. What about the external environment, the solvent?
The properties of the surrounding medium, especially its polarity or dielectric constant, can also significantly affect the pK.
How?
Think about dissolving a carboxylic acid, RCOH, RCO plus GO plus, in a solvent less polar than water, say, by adding ethanol.
Okay, lower dielectric constant.
Right. A lower dielectric constant means the solvent is less effective at stabilizing separated charges, like RCOONH+. It costs more energy to create those ions in a less polar solvent.
So the equilibrium shifts back towards the uncharged RCOOH form.
Yes. Less dissociation means it's a weaker acid in that solvent. So the pK of a carboxylic acid generally increases weaker acid in a less polar medium.
What about something like an amine RNH3 plus RNH2 plus H plus 2? The acid form is charged, the base form is neutral.
Good point. In that case, moving to a less polar solvent destabilizes the charged RNH3 plus form more than the neutral RNH2 form. This makes it easier to lose the proton. So the pK of an amine typically decreases stronger acid in a less polar medium.
So the effect depends on the charge states of the acid and conjugate base.
Exactly. And this becomes incredibly important inside proteins.
Why?
Because the microenvironment around an amino acid side chain within a folded protein can be very different from bulk water. It might be buried in a hydrophobic core, low polarity, or next to other charged groups, or have specifically positioned water molecules.
So the pKmi of a specific residue, like a histidine or aspartate, inside a protein can be quite different from its pKmi when it's free in solution.
Dramatically different, sometimes by several pH units. This fine-tuning of pKi values by the local protein environment is absolutely crucial for enzyme catalysis and protein function. It allows residues to be protonated or deprotonated at physiological pH in ways that wouldn't happen otherwise, enabling them to act as specific acid or base catalysts, for example.
Wow. So the protein structure itself manipulates the chemical properties of its own residues.
It's a key part of how proteins achieve their specific functions.
Yeah.
The interplay between water, pH, pKi, and the local environment is incredibly sophisticated.
We have certainly taken a truly deep dive into water and pH today. It's really quite astonishing how these fundamental concepts underpin so much complexity.
It really is. We've covered a lot of groundwater's unique properties, from its polarity and hydrogen bonding.
Making it an amazing solvent and shaping biomolecules through the hydrophobic effect.
Right. And the other non-covalent forces. We looked at its active role as a nucleophile in hydrolysis and group transfer,
Essential for metabolism.
and then dissected water's own ionization, the meaning of pH, the behavior of weak acids and bases, which is so critical for biomolecules.
Leading to the Henderson-Hasselbalch equation and the vital concept of buffering.
Exactly. And finally, how pKi values themselves can be tuned by the environment. Mastering these principles really does give you a powerful lens to understand biochemistry.
It really makes you think, doesn't it? Consider how just subtle shifts in water's local environment or the pH can cascade through the delicate machinery of a cell. It makes you wonder, what other molecules, ones we might think of as simple, hold equally profound secrets about how life, or even the universe, works?
A humbling thought. There's always more to discover.
Thank you for embarking on this deep dive with us today, exploring these foundational concepts, and thank you, our listeners, for joining us.